Hi guys, so just a brief recap of last time we talked about viral reproduction, how they do it. Um, so viruses attach to the surface of its host cell. An infection begins when the genetic material, either DNA and RNA, makes its way into the host cell. Once inside, the virus hijacks the cell and programs it to start making viral parts. The host cell will begin to make copies of the viral DNA or RNA and produce the protein capsids. Then the host cell assembles the parts into new viruses, and the reproductive cycle ends with the exits of hundreds or thousands of viruses from the infected cell. This often kills the host cell, and each of these viral progeny or the new viruses has the capacity to infect neighboring cells. That is not the whole story, though. There are two reproductive possibilities. Once a virus is inside a host cell, two different processes may occur. The first one, which is labeled number 2 here, some viruses replicate themselves immediately, killing the host cell. However, other viruses replicate themselves in a way that does not destroy the host cell, or at least not right away. These two processes, are called the lytic cycle or the lysogenic cycle. So let's talk about the lytic cycle. This is the one that kills the host cell right away. In a lytic infection, a virus enters a cell, makes copies of itself, and causes the cell to burst. Bacteriophage T4 is an example of a bacteriophage that causes a lytic infection. So let's look at how this diagram goes. This is a copy of the lytic cycle. Number 1, attachment. Tail fibers are used to attach to receptor sites on the surface of the host cell. So here we have a bacteriophage on top. It's got its blue DNA or RNA, and then it's got its tail fibers that attach to the bacterial cell via the bacterial cell's receptors. 2, entry. Phage DNA or RNA is injected. The empty capsid remains outside. The host cell DNA is destroyed. So the cell is going to die anyway because now it has no DNA of its own, but it has the blue viral DNA or RNA. Synthesis. The host cell is directed to produce viral genomes and protein capsids. So we're using the virus genetic material to make viral parts here. And then assembly. The viral DNA, went too fast. Sorry guys, assembly. The viral DNA or RNA is assembled inside the protein coat. So we've got new viruses packaged inside. And they're so crowded, they're going to finally release. The cell swells, bursts, and release hundreds of new viruses. Now we're going to end the video for now and talk about the lysogenic because our time's almost up for Screencastify, and join me back in just a bit.